Welcome everybody to a new global IMMUNOTALK. I'm Carla Rothin and I'm here with our co-organizer Elina Zuniga. We're delighted to welcome you again. I am looking forward to today's talk, and please join us next week also for the talk by Miguel Soares. So with this, I let Elina introduce our speaker today, Dr. Janelle Ayres.
Thank you, Carly. I'm delighted and super honored to introduce our speaker today, Dr. Janelle Ayres. Janelle earned her bachelor's degree from UC Berkeley in molecular and cell biology. She then went to Stanford School of Medicine for her PhD studies in the laboratory of Dr. David Schneider. And there, she used Drosophila to elucidate principles of host defense and host pathogen interactions. She there discovered that in addition to resistance mechanisms that are executed by the immune system and are important for pathogen killing, animals evolve a distinct defense strategy that she calls disease tolerance. And this is essential for the survival of infectious diseases and to promote cooperation between host and pathogens rather than antagonistic interactions. So this was really a breakthrough discovering host pathogen interactions. After a short postdoc in Russell Bunce's laboratory at UC Berkeley, Janelle moved to the Salk Institute in San Diego in 2013. And since then, Janelle has developed a multidisciplinary program using invertebrate and vertebrate models of host-microbe interactions that have allowed her to continue elucidating the mechanistic basis of disease tolerance and unravel important biological principles ranging from host defense against infections to evolution of commensalism. And I'm sure we will learn more about all this in her talk. So in 2016, Janelle was promoted to associate professor at Salk, and then in 2019, she was promoted to full professor. So she really scaled through the ranks at the Salk Institute. So Janelle's terrific insights into the field of host-microbe interactions have been recognized by many prestigious awards, including the Salk Scholar Award, the NIH Director Pioneer Award, and the Blavatnik Award for Young Scientists. Her work has been published in top-tier journals, including Cell, Science, Nature Medicine, and is also highly cited. On a personal note, I would like to emphasize that I feel really lucky to have Janelle as a colleague in the La Jolla immunology community. She has always been very generous to provide super valuable feedback in all our interactions as a member of thesis committees or grant review panels. And I am very, very grateful to have Janelle close by as a colleague. So I cannot wait to hear Janelle's global IMMUNOTALK, which is entitled Host Pathogen Interactions, Harnessing Co-evolution to Treat Disease. So Janelle, thank you so much for all the efforts to prepare this talk and for being our global immuno speaker today.
Thank you, Elena, very much for that introduction. And I'm very happy to be here, but I also want to say I'm very happy to have you as a colleague and to be in the same community as you. And I miss you. We haven't seen each other in a year, so you know, it's unfortunate this has to be virtual. But I want to thank you and Carla for making this happen and keeping the immunology community going during this time. This has been really great.
Thank you, Janelle. I appreciate the nice words. Thank you so much. And so we want to learn more about you. And so we have a question. And so as a scientific leader in the mechanisms that drive disease tolerance to infection, which is quite distinct from the most widely studied host defense response, and yet a fundamental component of the responses to infection, we would like to ask you if you could share with us what have been the opportunities and the challenges of taking this road less traveled.
Mm-hmm. Yeah, so I think there's of course many opportunities and many positives to taking the road less traveled. So I think one of the most important things we need to do as scientists is really examine the field and think about what are the unique perspectives and contributions that I could be bringing to the field. And so by finding disease tolerance and establishing this as a niche has enabled me to do so and to be able to make unique contributions to the host defense and host microbe interaction field. It's incredibly exciting to be thinking about things from a different perspective, and with that comes discoveries that haven't been made before, right? So this is just very exciting to be a part of, and to be a part of something from the very beginning. And the field, there's not many of us, but there's a really great group of people that are in this field, and so we're all distinct, but we are all very passionate about this concept, and all of us helping to move this field forward has just been really exciting. And then for the negatives, this was actually hard for me to think of something, because I think with everything that can feel as a negative experience, you can turn it into a learning experience in science, and then that becomes a positive. So of course, I learned at a very early stage of my career what it feels like to be told I'm wrong often, and that what I work on is not important, or people don't understand it. And that, of course, is something we hear often as scientists via peer review or various mechanisms. And it helps one develop a thick skin early in their career. But I think also it helped me to, rather than just be dismissive and say, oh, these people just don't understand what I'm saying, it helped me to listen to what they were saying, and to really help me refine the way I was describing things. And so I'm constantly listening and refining the ways in which I describe things. And I think that hopefully shows in the way we write about these concepts to help make it more understandable for others and better received. And I think it also helped me to see the importance of, if you want a field to move forward, you can be both critical and supportive. And I think that is an important thing that scientists need to do, is it's okay if a mechanism isn't perfect, but if you like the concept or the idea, you can still say you like something and be supportive of it to help the field move forward.
Nice. Perfect, Janelle. Thank you so much. Very helpful. Thank you for sharing your thoughts and for all your out-of-the-box contributions and perspective to science. Yeah, thank you so much. So without further ado, if you could share your screen.
I'm hoping that this goes smoothly. I was telling Carla and Elena about a very simple animation I had last week that tripped up my whole system. So I got rid of that animation. And hopefully this goes.
Perfect. We can see the slide perfectly well.
Okay, great. So let me just hide all of you. Okay, great. So again, I want to thank Elena and Carla for the invitation to be here today, and for all of you for joining in. And we, in my lab, we study everything host microbe interactions, both pathogenic interactions as well as commensal or mutualistic interactions. And really the question that motivates my research program is, I think it's a deceptively simple question, and it's basically, I want to know if an individual contracts an infection, what dictates that individual is going to survive the infection, or if they're going to become sick and die from the infection. And so we've turned to a nearly 100-year-old biological phenomenon to help us answer this question. So lethal dose 50, or LD50, is the dose of a microbe that will kill 50% of a genetically identical host population, while the other 50% survives. And here I'm showing you the death kinetics of a group of mice that have been challenged with a natural mouse pathogen called Citrobacter rodentium. And this is related to the pathogenic E. coli that can infect and cause disease in humans. So this infection in mice, it causes infectious colitis, and it's characterized by colonic damage and inflammation. And importantly for our purposes, this infection, it causes mice to present with predictable clinical signs of disease, including an overall morbidity. There's changes in their stool consistency, so they develop diarrhea, and they also develop an overall wasting phenotype that's characterized by the depletion of both lean and fat energy stores. And most importantly for our purposes, these clinical signs are quantifiable. And so what that means is we can go in every day post-infection, and we can clinically assess animals based on these different criteria and assign them a score to quantify disease severity. And with those clinical scores, we can then generate health trajectories of the animals over the course of the infection. So for example, if we take animals that have been challenged with the LD100 dose of Citrobacter, so 100% of the animals will succumb to this infectious dose. When we, what we see with the health scores and the health trajectory is that the health scores will decline for every animal over the course of the infection. So that indicates that every animal is getting sick in response to the infection. And this is exactly what we would predict to happen, right? We would predict if we infect an animal, they're going to show signs of sickness, and they're going to ultimately succumb to the infection. And we would predict that their health trajectories would look like this, that it's going to continue to decline over time. And if they ultimately succumb to the infection, that the trajectory will continue to decline over the course of the infection until termination. But the question then is, what would we predict the health trajectory of the survivors of the LD50 to look like? And I think most would predict, well, the survivors of the LD50 should also exhibit signs of sickness, so their health trajectory will decline. And at some point, there will be an inflection point in the curve, and their severity of sickness will level off, and then there would be an additional inflection point, and the animals will then establish their route to recovery and hopefully return to a baseline health phenotype. And this is what we would call a resilience health trajectory. But when we applied our clinical scoring system to the animals that were challenged with the LD50 dose of Citrobacter and looked at the health trajectories of the animals that fell into the two different fate groups, we revealed something that I found to be very different and very exciting. And what we found was that there was a bifurcation in the health trajectories of the animals that fell into the two different outcome groups. So the animals that would ultimately succumb to the infection, they became sick and their health trajectories declined, as we would predict. But the animals that fell into the surviving outcome group, they were able to maintain their health trajectories at maximal health over the course of the infection. And so rather than exhibiting a resilience health trajectory, the survivors of the LD50 exhibited what I call an endurance health trajectory. So they were able to maintain maximal health after receiving the infectious challenge. And I think most people, if they were faced with these data, they would suggest that, well, perhaps that there was just some stochastic event that occurred, and that stochastic event led to differences in pathogen burdens in the two different outcome groups, and that the sick dying animals are sick and dying because there's really high levels of pathogen, and the healthy surviving animals are healthy and surviving because there's low levels of the pathogen, and that the host outcome of infection is a direct function of pathogen burden. And so we wanted to, of course, test this idea in our LD50 system. And so to do that, we harvested the target organs of Citrobacter at day 8 post-infection, when the sick animals reached maximal morbidity, and we quantified pathogen burdens. And what we found was that the differences in host health and outcome in animals challenged with the LD50, this could not be explained by differences in pathogen burden. So we find comparable levels of the pathogen in the animals that become morbid and will ultimately succumb to the infection, and those that remain healthy and survive the infection. And so this indicated to us that the host is capable of changing its relationship with the pathogen so that health and survival is achieved rather than disease and death. And this is what we're trying to understand in my lab mechanistically so that we're better able to treat diseases. And I think most investigators, if they were faced with these health trajectories, they would focus on understanding the mechanisms that shift the health trajectories downward, because a common assumption in biomedical research is if we can just understand the mechanisms of disease pathogenesis, then we should just be able to block those pathways, and that should be sufficient to restore a sick, dying individual up to a healthy, surviving individual. And while that's important and has proved to be a beneficial way of thinking in biology, we're thinking about this from a different perspective. And we're proposing that the mechanisms that enable the healthy surviving mice to stay healthy and survive during the infection are distinct from the mechanisms of disease pathogenesis. And this is what we're trying to understand in my lab so that we can learn how to shift the health trajectories of these sick individuals up towards health during an infection or a challenge. So now, how is this even possible? How can you have a host that's received this infectious challenge? It has high levels of the pathogen present in its body, but it's perfectly healthy and showing no signs of disease. And so we've spent more than a decade now developing a conceptual framework to help us understand such phenomenon. And the way I like to think about host defense strategies against infection is I think about them in this health by microbe space. So we have health of the host on the y-axis and pathogen burdens on the x-axis. At the intersection of the axes, that's uninfected, and the host is at maximal health. And then along the x-axis, as we move along it, pathogen burdens increase. And so what we do is we think about how the host moves within this space and how that movement relates to health and pathogen burdens of the individual. So when they're uninfected, they're at maximal health and they don't have pathogen present in the body. When the host becomes infected, pathogen levels can increase and health of the host will decrease, indicating that they become sick. And so one of the ways in which the host can move back to the uninfected or healthy state or prevent sickness from even happening is they can promote or they can employ antagonistic defenses that protect the host by killing or eliminating the pathogen. And so these defense strategies are going to be reflected by shifts along the diagonal to indicate that host health is the function of pathogen burden. So the lower the amount of pathogen, the healthier the host is going to be, and the higher the amount of pathogen, the sicker the host is going to be. So antagonistic defenses are very familiar to us. Of course, these are mediated by resistance mechanisms that are encoded by the immune system that are essential for destroying and eliminating pathogens. But unfortunately, a consequence of these antagonistic defenses is that pathology can occur. And this can be due to direct effects of the pathogen on host physiologies at all scales, but it can also be due to the nonspecific nature of antagonistic defenses. So for example, one of the pathologies that we study in my lab that's common to both infectious and non-infectious diseases is called muscle wasting, or it's called cachexia in the cancer world. And so this is defined as the loss of lean energy stores that occurs during disease. And one of the pathogens that we use to induce muscle wasting in our animal models is called Burkholderia. So we can administer this pathogen intranasally to mice, and they develop a pneumonia-like disease. And one of the pathologies that they exhibit is the animals will develop this very rapid and robust whole body wasting phenotype. And we can do MRIs on the animals. We can also weigh individual muscle stores in these animals, and what we see is that the the whole body wasting can be explained by muscle wasting that's occurring in these animals. And we can confirm that muscle wasting is happening because when we look at the induction of the muscle atrophy machinery in these animals, we see that it's being induced during infection. So in this infection setting, the muscle wasting is being caused by the host response to the infection. And so, so the host immune response to the infection. So because wasting and other pathologies that can occur during infection can compromise both host and microbial survival and fitness, we propose that a host can also defend against infections by adapting to the presence of the pathogen. And this can be done to achieve resilience or an endurance health trajectory. And I suggest that this can yield an apparent cooperation between the host and the pathogen. And this can be achieved by the host employing what we call cooperative defenses. And so these are important for protecting the host by preventing physiological damage during infection and without killing the pathogen. And these defenses, they would be reflected by shifts along the y-axis, indicating that host health is a function of how well the host can adapt to the presence of the pathogen. So in support of this, more than a decade ago, when I was in David Schneider's lab using a Drosophila model, as Elena told you, we described that animals encode a distinct defense strategy that we call disease tolerance. This is absolutely essential for an animal's ability to survive microbial interactions, and it protects the host and promotes host health while having a neutral to positive impact on microbial fitness. So unlike resistance mechanisms, which protect the host by killing the pathogen and having a negative impact on pathogen fitness, disease tolerance can promote health without killing the pathogen. And so importantly, disease tolerance does not affect the ability of the microbe to engage the effector pathways that lead to disease pathogenesis. And instead, these mechanisms work in parallel to induce health in or maintain health in the host. And these mechanisms, they operate across all scales in the body, so cellular, tissue, organ, and physiological, to limit susceptibility to pathogenic signals. They can mediate function of these systems should damage occur, and they can also mediate repair should damage occur. And I just want to point out that Andrew Reed and Lars Rådberg at the same time, they had demonstrated in a mouse model of malaria that disease tolerance exists in invertebrates. So in an infected host, when these mechanisms are then activated, they can promote health despite the host being infected with the pathogen. And so just to give you an example of a disease tolerance mechanism, so a few years ago now, we had published our discovery of a commensal, this E. coli O21, where we found we could basically administer this E. coli as a probiotic to animals that were infected with Burkholderia. And if we orally administered this E. coli, it would colonize the intestines of the animals, and that was sufficient to protect the animals from Burkholderia-induced muscle wasting. And importantly, when we quantified pathogen burdens of Burkholderia in the lung, we found that there were no differences in the animals that received this E. coli probiotic and were protected from Burkholderia-induced muscle wasting compared to the animals that received the vehicle and were infected with Burkholderia and exhibiting muscle wasting. And so what this demonstrates is that intestinal colonization with this E. coli was not protecting the animals from Burkholderia-induced muscle wasting by heightening the host immune response to the pathogen. And it also demonstrated that this E. coli was not competing for a niche with the Burkholderia. And so that suggested to us that this E. coli was promoting a disease tolerance mechanism, protecting the animals from developing muscle wasting without having a negative impact on Burkholderia fitness. And so we used that E. coli to elucidate a disease tolerance mechanism. And consistent with this being a disease tolerance mechanism, we found that intestinal colonization with this E. coli did not affect the ability of Burkholderia to induce the upstream positive regulators of muscle wasting. And instead, what we found was that it was inducing a parallel pathway that, that changed the sensitivity of the skeletal muscle in such a way that it did not experience wasting during the infection. So consistent with this being a disease tolerance mechanism. So now going back to our LD50 systems, we had hypothesized that perhaps the healthy infected animals in this LD50 Citrobacter system, they were healthy despite infection because there was the induction of disease tolerance mechanisms in these mice, and shifting this host pathogen system from one of antagonism where disease occurs to one of cooperation where both host health and pathogen fitness can be achieved. And so to test that, we took animals, we challenged them with the LD50 dose of Citrobacter, and then we applied our clinical scoring system to the animals. And when the sick dying animals reached maximal morbidity, we harvested organs from these animals. What's relevant for my talk today is the liver, and we also harvested livers from uninfected healthy control animals. And then we performed RNA-seq on the livers, and we specifically were looking for pathways that were induced specifically in the infected healthy individuals compared to both infected morbid, but also uninfected control animals, because those are going to be the genes and the pathways that would most likely reflect adaptive mechanisms that are important for promoting health during infection and support our hypothesis that there was the induction of disease tolerance defenses in infected healthy populations, enabling them to stay healthy while maintaining pathogen fitness. And so when we looked at the data, we found consistent with our hypothesis genes that were specifically upregulated in the infected healthy mice compared to both the infected sick or morbid and the uninfected healthy animals. And rather than revealing the expected immune and inflammatory functions, we found that these genes represented various aspects of host metabolism or the endocrine response and other non-immune and inflammatory functions. So that was exciting to us, but of course we wanted to know if we could use this information that we were revealing from our LD50 systems to understand mechanistically how we can shift a host pathogen system from one in antagonism to cooperation and shift health trajectories of animals up towards a maximal health and an endurance trajectory. And so when we went back to our RNA-seq data set and we were deciding what to go after, we were intrigued by what we had pulled out in this mineral electrolyte absorption category. And so what we had pulled out of that was various genes that are important for host iron physiology. And so that was oh, duplicate slide. So that was interesting to us because of course, iron has a very well appreciated role in host pathogen systems. And the traditional way we think about this is that changes in host iron physiology that occur during infection, these are reflective of antagonistic defense strategies. And so one of the ways that we're very familiar with is during infections, the infection will trigger a fever response and acute phase response. And this can lead to changes in host iron metabolism, including cellular and tissue iron sequestration of iron. And we say that these changes reflect antagonistic defenses because pathogens, of course, many of them thrive on iron. And so by removing or reducing the amount of circulating iron, that would have a negative impact on pathogen fitness because we say it's essentially starving the pathogen of iron. But our data suggested with our LD50 system that perhaps these changes could be reflective of a cooperative defense strategy, at least in this host pathogen system. So to test this idea, we took a dietary approach. So we took animals and challenged them with the LD100 dose of Citrobacter and gave them control chow. So 100% of the animals will become sick and succumb to the infection. Or we gave a group of mice that were infected with the LD100 dose a 2-week course of chow that was supplemented with a non-toxic dose of dietary iron. And then after that 2-week course, we removed it and put them on normal chow. And we found that if we just gave the animals this 2-week course of dietary iron, that was sufficient to confer 100% protection in these animals. So we can shift what should have been an LD100 to an LD0 just by giving this common dietary element to these animals. And we wanted to push the system as far as we could technically go. So we challenged the animals with up to 1,000 times the LD100 dose. And just by giving a 2-week course of dietary iron, that was sufficient to confer 100% protection, even at these really high doses of the pathogen. And so in addition to promoting survival, we found that giving the dietary iron was sufficient to promote an endurance phenotype in the animals, so they were able to maintain their health at maximal health after the infection. And when we quantified pathogen burdens, we found that there were comparable levels of the pathogen in the iron-treated group as well as the control-treated group. And so what this demonstrates to us is that the dietary iron is not promoting health and survival because it's promoting antagonistic defenses, but rather it's promoting cooperation between the host and the pathogen, where host health can be maintained and pathogen fitness can also be maintained. So we used this system to define a cooperative defense mechanism. And so to help you understand how we went about this, I have to tell you how Citrobacter causes disease in its host. So when Citrobacter reaches the colonic niche, it will induce expression of various virulence factors that are harbored by a pathogenicity island, which is a cluster of genes on its chromosome that encode virulence factors that mediate disease pathogenesis. So when it expresses these virulence factors, it enables it to attach to the colonic epithelium, and there's rearrangements that happen in the host cell, and that leads to the effacement of the colonic epithelial cells, and that results in the inflammation, the colonic damage that contributes to the clinical signs of disease that we use for our system. So when during the infection, as I said, these virulence factors are transcriptionally upregulated, and so in our control diet setting, we see the expected transcriptional upregulation of these virulence factors. But if the animals are given the dietary iron during the infection, we are able to prevent transcriptional upregulation and expression of the pathogen's virulence program. So it seems that what we have created was asymptomatic carriage or colonization by this pathogen. It looks like it's basically existing as a commensal in these animals. And so we considered various, we went down various lines of investigation to understand how this could be happening, but ultimately our analysis led us to consider the relationship between host iron metabolism and host glucose metabolism. So iron affects various metabolic organs and tissues in mice and in us, and can affect glucose metabolism and homeostasis. So what's relevant for our purposes is the effects of dietary iron on white adipose tissue. So this had already been reported to cause declines in adipose tissue mass and lipolysis, and the white adipose tissue can sequester iron, and also insulin resistance in white adipose tissue. And so consistent with that, when we looked in our infection model, we found that the animals that were infected and received the dietary iron, they had increased accumulation of iron in their white adipose tissue depots. And also consistent with previous work, we found that the animals exhibited a reduced percent body fat if they received iron during the infection. And so what we found was that the animals had developed systemic insulin resistance, and we can, so this is an ITT, or an insulin tolerance test, where you inject animals with insulin and you look for the rate of reduction of glucose in the circulation. And you can see that the animals that received iron during the infection, they are more insulin resistant compared to the infected animals on the control chow. And when we look at insulin signaling in white adipose tissue, we see consistent with the ITT phenotype, we see that there's reduced insulin signaling in the white adipose tissue of animals that are infected and receive the iron chow. And consistent with this being an insulin resistant phenotype, when we look at circulating levels of insulin, we do not see any differences in the two dietary conditions during infection. So this tells us it's a resistance phenotype rather than differences in insulin secretion or circulation. So I'm just going to sum up this part for the interest of time. So basically what we found was that, and what I've told you thus far, is that if the animals are given control chow during the infection, they're insulin sensitive, and if the animals receive dietary iron, they develop this transient state of insulin resistance. And we showed that this transient state of insulin resistance was necessary and sufficient to confer protection against Citrobacter, and that it was necessary for iron-mediated protection against Citrobacter disease and induced disease and lethality. And what we also found was that this transient insulin resistance was increasing, it was decreasing glucose absorption and increasing the amount of glucose that was available in the intestinal lumen. And so the next step was then to ask, well, what is the importance of this increased availability of intestinal glucose for iron-mediated cooperation between the host and Citrobacter? And so to test for necessity, what we did was we employed acarbose, and so this is an alpha amylase and alpha glucosidase inhibitor that will prevent the liberation of glucose from diet. And so if you look at the blue line, what we found is that if we infect animals with citro, we give them the dietary iron, and if we also treat them with acarbose to prevent glucose administration from the diet and decrease the amount of glucose availability in the intestinal lumen, now the animals are susceptible to a Citrobacter infection despite receiving the iron chow. And so that tells us that this increased intestinal glucose availability is necessary for iron-mediated suppression of Citrobacter-induced disease and lethality. And to address sufficiency, we took animals that were on control chows and did not receive iron, and we challenged them with oral boluses of glucose, and we found that that was sufficient to protect animals from Citrobacter-induced disease and lethality. And importantly, this administration of glucose had to be done orally. If we administered it systemically, we did not see any protective or detrimental effects of the glucose administration. So then the question is, why is this increased glucose availability in the intestine? Why is that necessary and sufficient to suppress Citrobacter-induced lethality and disease? And the reason we found is that when glucose is the carbon source for Citrobacter, it prevents the pathogen from engaging its virulence program. So it's able to still colonize the niche of the pathogen in the host, and it can replicate, but it no longer engages its virulence machinery. So it is essentially existing as a commensal in the host. And so just to sum up what I showed you in our model thus far again, so if the animals are infected and given control chow, they're insulin sensitive, this increases glucose absorption from the gut. That reduces the amount of glucose availability in the intestine for the pathogen to forage on. That triggers pathogen virulence, and we get disease and death of the host. If the animals are given dietary iron, they develop this transient state of insulin resistance. This reduces glucose absorption from the intestine, and that increases the amount of glucose availability in the intestine for the pathogen to forage on. That suppresses pathogen virulence, and they basically become asymptomatic carriers of the pathogen. So we've promoted cooperation in this host pathogen system. But when we were finished with this project, we we asked this important question: did we actually reveal a disease tolerance mechanism with this work? So I told you earlier that disease tolerance does not affect the ability of a microbe to engage the pathways that lead to disease pathogenesis, but rather they induce parallel pathways that work to promote health. But what I just showed you with our iron and our insulin resistance work is that iron and insulin resistance and glucose availability, they're acting here to basically suppress the induction of the pathogenic pathways that are employed by the pathogen to even cause disease. And so what this looked like to me was that the dietary iron and the insulin resistance and the glucose availability, they were essentially acting as an anti-virulence mechanism. And so I think what that indicates is that we've discovered a new strategy by which a host can promote cooperation with the microbe. And of course, it's well established that pathogens have evolved anti-virulence mechanisms that are necessary to regulate their own virulence. But what we've demonstrated is that the host has also evolved physiological responses that can act as anti-virulence mechanisms to enable the host to maintain health while maintaining pathogen fitness. And what I think it looks like actually is that the host is bribing the pathogen. So the host is saying, you know, I'll give you this nutrient source or a stable niche for acquiring nutrients and for replication and transmission if you don't cause disease in me. And bribery it's a very familiar tactic in other cooperative to maintain cooperation and other symbiotic systems. And I think the classic example of this would be the ant-aphid system, where the aphid feeds the ant a sugary substance called honeydew. And in doing so, it essentially bribes the ant into not attacking the aphid, but it also bribes the ant into sticking around to provide defense for the aphid against other enemies that can come into this system. And so I think we've revealed something that I like to call metabolic bribery, in that metabolic bribery can basically serve as a cooperative defense mechanism to also mediate cooperation in host microbe systems. And I think that there's likely more that are important for this metabolic bribery. And there are other examples that have been discovered, and I think that there's many more to be discovered, and they're likely context dependent, depending on the particular host microbe interaction and the context in which those interactions occur. So when we finished this, we had to really expand our view of the strategies by which a host can cooperate with microbes. And that in addition to disease tolerance, hosts have evolved what we call anti-virulence defenses. And both of these operate to limit physiological damage during infection without compromising pathogen fitness. And we say that both anti-virulence and disease tolerance make up what we call the cooperative defense system. So this is distinct from the immune system, but complementary and very important component of the host defense response to infection. And so that's just one example that to demonstrate how we use these LD50 systems in my lab to understand disease tolerance and anti-virulence defenses. And so we have expanded our efforts significantly and developed LD50 systems for various different infection models that are pathology specific, but also models that have common pathologies to each other. And this has launched my lab's LD50 program, where we're interested in leveraging these systems for various things for the disease tolerance field, where we, of course, want to use these systems to continue to define mechanisms of cooperative defenses. Something that I'm really interested in is understanding what even dictates or what defines the LD50 in the first place. So what dictates the trajectory an individual is going to take. Again, these are genetically identical animals. They have the same microbiota compositions. They're in the same cage environment. So what is different that's even dictating these trajectories? We also are using them to identify predictive markers so that we know the trajectory an individual is going to follow before they even show symptoms or signs of disease that they're going to. And then we use those predictive markers to inform us on methods of intervention so that we can shift trajectories up. And we also want to understand the evolutionary and epidemiological implications for cooperative interventions. So if we promote disease tolerance or antivirulence in a host pathogen system, what are the evolutionary and epidemiological implications for that? And that's what I want to spend the remainder of my time telling you about how we're thinking about this today. So I showed you that promoting cooperation between a host and a pathogen is beneficial for an individual host because you're promoting health and survival of that individual host. And so that, of course, is very good for that individual host, but it raises an important question of how beneficial is this for a community or a population level, because we're potentially just creating a case of typhoid Mary or other asymptomatic carrier type scenario where the individual is healthy, but now you're creating a scenario where that individual can go into the community and can spread a virulent pathogen to naive individuals within that community. And so is this even a good idea, or is this going to be bad? And I think it still is a good idea. And I don't think that we're always going to be creating typhoid Mary scenarios. And so I will explain to you my logic for that and then experimental evidence to support the logic. So with antagonism, a pathogen is going to infect a host population, and it's going to employ adaptation factors that operate to promote pathogen fitness by causing disease in the host. And so that's going to drive the selection for resistance defense traits in the host population. And that's going then, of course, to protect the host by killing the pathogen. And then that's going to drive the selection for new or different antagonistic traits in the pathogen population that promote pathogen fitness when infecting the host by causing disease. And so this, of course, it's very familiar to us. We often call this the arms race. It's more accurate to call it the Red Queen effect, where there's the oscillation of antagonistic traits in both the host and the microbial population. And this is necessary in order for each party to maintain their fitness status. So for example, virulence factors, basically all those are adaptation factors that operate to promote pathogen fitness when infecting the host niche, and they do so by causing disease in the host. Now, in the case of cooperation, you'll have a pathogen that infects the host population, and this will lead to the selection for disease tolerance and antivirulence defense traits in the host. And then those traits, of course, are going to protect the host by providing physiological defenses and preventing physiological damage. This will have a neutral to a positive effect on pathogen fitness. And so the important implications of that is that they theoretically are not going to provide the same selective pressures that antagonistic defense traits do. And so therefore, we would predict that these are not necessarily going to drive the selection for new or different adaptation factors in pathogen populations that are going to operate to promote pathogen fitness by causing disease in the host. And so we've taken this a step further to suggest that if microbial fitness can be maintained through the maintenance of host health, then theoretically there can be the selection for strains that lose virulent potential, so long as the pathology being alleviated by the disease tolerance or the antivirulence trait is not needed for transmission or replication. And so we can test this idea with our LD50 systems and with our systems where we've established where we can intervene and promote cooperation in the host pathogen system. And so we can take, for example, our dietary iron system, where we take what should be a antagonistic host microbe system. We can give them the dietary iron for a couple weeks, shift that individual host pathogen system to one of cooperation, where host health is achieved and pathogen fitness is maintained. And we can ask in this system have we created an environment where we drove the selection for attenuated strains of the pathogen. So I'm taking you back to the original experiment we did, where we challenged animals with 1 to 1,000 times the LD100 dose of Citrobacter. We gave the animals dietary iron for 2 weeks, after which we withdrew the dietary iron and we put the animals on normal chow for the rest of their lives. So this is fast forwarded about 18 months post-infection, and the animals that were infected with Citro and given the 2-week course of dietary iron, they're still alive. Clinically, they look fine. They look the same as the uninfected control animals we have in parallel. And importantly, when we look at the feces of these animals, we see that they're still shedding Citrobacter. So what that demonstrates to us is that this short-week course of dietary iron provides long-term suppression of Citrobacter virulence. So this long-term suppression of virulence, it could be a phenotypic suppression of virulence. So they're phenotypically attenuated, but if we take them out and move them into a new host, they're going to be virulent again. Or it could be that we've driven the evolution of attenuated strains. So we can test that by isolating Citrobacter from the feces of these animals and then infecting naive animals with control chow with these isolates and ask what happens. And so when we do that, we see that that the parental animals infected with the parental strain and on control chow, they succumb to the infection as we would expect. But the animals that receive are challenged with one of these different isolates that we isolated from long-term carriers of Citrobacter that receive the dietary iron. They do not cause lethality in the animals. They don't cause disease in the animals. And importantly, when we look at the feces of these animals that were challenged with the isolates, we see that they're still harboring the Citrobacter isolate. So it's capable of colonizing them, it's capable of persisting in them, but it doesn't cause disease, and they don't cause lethality. So that demonstrates to us that this short course of dietary iron drove the selection for attenuated strains of Citrobacter. And when we sequenced the genomes of these isolates, we found consistent with the phenotype we observed that these isolates, they had acquired various mutations in the genes harbored within their pathogenicity islands. So various mutations in genes that encode virulence factors, rendering them non-functional. And so at least in the case of Citrobacter and iron and in mice, we demonstrate that this is not only good at the individual level, but it could be good at the community level. And we don't know if all microbes and all cooperative defense mechanisms are going to follow these rules, but we would predict that any microbe that follows these rules will always trend toward avirulence, provided that the virulence and the disease and pathology that can be triggered in the host is not needed for replication or transmission. And I would suggest that unless a microbe is 100% lethal, any survivor can act as a source of avirulent strains. And so we're using our LD50 systems so that we can understand all of the principles that govern these relationships. So are all host microbe systems going to trend and methods of cooperation and promoting disease tolerance and antivirulence, will they always trend towards avirulence? Are there scenarios where we can actually push them out the other way and they can become hypervirulent? Are there situations when we can drive attenuation, but then they revert back to the virulent state? So we're working to understand all of that in our systems. And of course, as we identify systems where we can drive the evolution of attenuation, we want to understand the mechanism of attenuation. So with our Citrobacter model, Grishka Chen, a postdoc in my lab, he's really interested in this question of understanding what is actually driving the attenuation. And what he has found is actually that the adaptive immune system is necessary for attenuation of Citrobacter in mice that are treated with iron. And so his studies thus far suggest that an antibody response is required for attenuation in mice that are treated with iron. And so this has led us to develop a multi-stage host defense model for the evolution of commensalism. So within this this mouse Citrobacter iron system. In stage 1, we have suppression of virulence, and this is dependent on iron and insulin resistance and our glucose phenotype, where iron triggers insulin in white adipose tissue that leads to systemic insulin resistance. This signals to the gut, it reduces glucose absorption, that increases the amount of glucose that's available in the intestine for the pathogen to forage on, and that suppresses pathogen virulence. So we have phenotypically avirulent as the insulin resistant wears off, we start to develop a mixed population, but we start to develop a phenotypically virulent population. And we get an IgA-mediated selective exclusion of these phenotypically virulent members of the community. And then we would predict that the IgA acts as the selective pressure to drive the evolution of the phenotypically avirulent members of the community to actually evolve to become genetically attenuated. And so what this, you know, I had always said prior to this project that antagonistic defenses are going to lead to an arms race or a red queen effect, and that it's cooperative defenses that are going to be relevant for driving the attenuation of a pathogen and evolution of commensalism. But what our data suggests is that really antagonistic and cooperative defenses are essentially cooperating with each other to promote the evolution of attenuation. And so that cooperative defenses are important for phenotypic attenuation and metabolically satisfying the pathogen in our system, and then basically enabling the animal to survive long enough for the antagonistic response to come in, and that provides the selective pressure for the evolution of attenuation. And so we're working on this, understanding the mechanism further, but of course, as I mentioned, understanding the rules and principles in our other systems, and if we're going to reveal similar things, or if we're going to find distinct things for different infection contexts, as well as methods of intervention to promote cooperation. And so just to end my talk, I'll put up this slide again just to remind you about animals and hosts have evolved 2 strategies to defend against infection in addition to the immune system, which is necessary to eliminate and kill pathogens. We also have what we propose to call the cooperative defense system, and this involves antivirulence defenses and as well as disease tolerance defenses, both of which are necessary to enable the host to maintain health or to recover from sickness while maintaining pathogen fitness. And so I'll thank all the members in my lab, but specifically I want to thank Karina Sanchez and Grishka Chen and Alex Schieber and Sam Redford. And then former member Michelle Lee, who has since graduated, they contributed to the work I told you about today. Various funding sources, and all of you for your attention. I'm happy to take questions now.
Thank you, Janelle. That was a terrific talk. It's always so enjoyable to hear your way of thinking and your different perspective. I really enjoy it. Thank you so much for sharing. And Carly will now direct questions to Twitter.
Yes, thank you.
Okay.
Thank you, Janelle. I mean, it's really impressive to see how you apply your creative thinking to fundamental biological questions. And it's a delight to learn from your studies.
Oh, thank you. You two are always so flattering.
No, this is really outstanding work.
Thank you.
So I'm sure people have questions. So just to remind them, they can go to the Global IMMUNOTALK account in Twitter, and they'll find already a tweet there that says ask questions for Janelle here. Please remember to add the hashtag #GlobalImmuno as you ask your question, and also refer to the Ayres Lab, that is the account that Janelle is going to use to answer her questions. So thank you, Janelle, again. It was wonderful to learn about your work. And thanks, everybody, for joining. And we look forward to seeing you next Wednesday. Thanks, everybody.
Thank you. Bye.
Bye. Bye, Janelle.
Bye. Thank you.